reminders about the lab quiz is opening up right at 1, and you'll have until next Wednesday. Okay. So, and then in Wednesday's lecture, I'll go over some of it and have a little bit of time during Wednesday's lecture to, if you need to ask questions about the exam content, so the lecture exam is next Friday. On Wednesday's lecture, I'll provide a little bit of time in case you have last minute questions, okay? So on the syllabus, it does state exam 1 is covering chapters 1, 3, 4, 11, and 14, which I'm covering 14 right now. And that should take care of it. So 1, 3, 4, 11, and 14. So there'll be a mix, multiple choice, true, false, that type of thing, and then at least one essay question, if not more. I haven't finished making it yet, so I will know more by next Monday. This Monday, 3 days from now. Questions on the exam right now? Why don't we do an iClicker? See if I can get that to work. Still, let me go. Whoops, not that. Okay, it should be open now. Ten more seconds. Okay, let's see what we got. Do it this way. Okay, so 93% said B, endophytes, and that is correct. Okay, so endophytes. Endo means within, and okay. So endophytes, the more they look at them, the more they realize they're doing all kinds of good stuff for the host plant. And so with endophytes, they're starting to realize, and sometimes the interaction is so tight that if you purge an endophyte out of the plant, it can't survive its habitat. So for example, they did experiments where this plant seemed adapted to really warm conditions. But what they realized it was that it was a fungus inside the plant that was allowing the plant to tolerate such high temps. When they got rid of that endophyte fungus, the plant did not survive. And so these types of things, they've done lots and lots of studies where they're showing that the presence of these endophytes are beneficial. So usually when you look up the definition of an endophyte, they typically will say something to that effect, that it is a beneficial interaction and that it's the case where it's either a bacteria or fungi. Viruses are probably playing a role too, but they haven't really got into viruses. So it's mainly they think about bacteria and fungi living inside the plant for most of their life and not causing disease is usually how they define endophytes. And the other thing that they're starting to realize is that a lot of the chemicals that we extract from plants, they're realizing it might be the endophytes that are actually doing that, which kind of puts a whole wrench in things in a way, because up until they knew about endophytes, they just assumed plants themselves were producing all these chemicals, and they sure do produce a lot of chemicals. That is not up for debate. They certainly do. But now we realize that the endophytes can also produce chemicals. Sometimes it's the exact same chemical the plant can make, but very often it's a unique chemical that the endophyte is for, in order to help the plant. And so when you do these chemical extractions, they're realizing it might be endophyte origin, really. And they're realizing endophytes can influence the production of secondary metabolites by the plant. And so they've done studies in many, many different plants, including cannabis, showing that the presence of certain endophytes can alter the ratio or increase the amount of certain secondary metabolites. So if you ever get into a job where part of your job is looking for, these secondary metabolites in the plants, keep in mind the idea of endophytes. I think a lot of businesses out in the real world really don't know about that. And so they're kind of working blind when it comes to that concept. And I think it could be a very powerful tool if you can understand it and work with it. It can be a very powerful tool to getting the plant to do what you want it to do. And in the case of businesses, make a profit off of it. So keep the endophytes in mind. They help protect the plant from pathogens. They can help against adverse environmental conditions. They ramp up key secondary metabolites. So we're discovering they're very, very involved in the plant's life. And so it's kind of analogous to what they're finding in the human microbe world, all the microbes inside of us as well. Questions on endophytes? You'll see a documentary as part of the lab component of this class on endophytes, which is really, really good. Get you thinking. But okay, so let's shift into what we were covering. Anytime, I tell you. All right. Let's see if I can get it to work. All right. So I think we left off here looking at this idea of using Agrobacterium tumefaciens as a tool to manipulate the kinds of genes that you want to see in that plant. And so the fancy word is transformation. When you insert DNA into some kind of organism, we call it transformation. And Agrobacterium tumefaciens is a plant pathogen that causes crown gall disease. And there's plenty of examples. If you've been out hiking around there, it's around. You see that large tumor-like growth on tree trunks in particular. That's usually Agrobacterium. And so what it does is it infects usually where there's a wound on the plant. So if there, something came along and gashed the tree trunk, the bacteria can get in, and then they start inserting their plasmid, which is an extracellular piece of DNA that they carry into the plant chromosome. And on that plasmid are genes to tell the plant grow, grow, grow. It's actually genes that code for auxin and cytokinin. And so those are the two hormones. I don't know, I can't remember if I wrote those down last time. Doesn't want right from here. So auxins, which you've learned about, and cytokinins. So these two hormones, the genes that code for those, are on that TI plasmid. And so those are telling the plant in that, let's say it's the middle of the tree trunk, it's telling those cells grow, 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 but don't differentiate. And so they grow into that tumor-like mass. And it creates a home for the microbes. They live in that mass. And so if you were to take a little chunk of that and look at it under the microscope, it'd be chock full of the agrobacterium. So it's telling the plant, make a home for me. And then it also carries a gene to tell the plant to make a nitrogen-containing compound that the plant can't use. And so it's telling the plant, okay, now make this nutrient for me, and it's only for me. You can't use it, but you get to make it. And so it's kind of a slick lifestyle for agrobacterium. It gets a home, it gets free food, and usually the plant does not die from this. And so again, agrobacterium, why would you want to kill your host if it's doing all that for you? You know, you get this free home, you get this nitrogen. And so the plant usually survives. But when they figured out that agrobacterium can insert its plasmid into the plant chromosome, I mean, that was big news. That was huge in the plant world. And so then, of course, they said, wow, why don't we take agrobacterium into the lab? Why don't we see if we can put our gene of interest into that plant? Maybe it's for drought resistance to help the plant withstand drought. And let's put that gene in there. And then let's take some plant cells. Maybe they want to work with the potato plant. And so some potato cells, mix it in with bacteria in a broth. And what happens is you get the situation where you can add all of this like into a flask. That's what they're showing right here. Okay, you add the bacteria, you take some plant tissue, mix it all together, and over time, what happens is the bacteria gets into those plant cells and does its job of inserting the plasmid, including the drought resistant gene, into the plant chromosome. So you have to do it in tissue culture like this. If you want every cell in the plant body to be transformed, you have to do it this way. You can't work with a mature plant. And so here what you do is you usually incorporate either an antibiotic also on the plasmid, and then, or in this case, they chose to work with herbicides. You need to be able to tell which cells took up the plasmid, which didn't. And so it's a lot like a marker gene that you put in there. And so here, we let the experiment go for a while, and then we made sure that on that plasmid was herbicide resistance as well, and drought resistance, let's say. And so then they added the herbicide, and they said, oh yeah, okay, the ones that survived got the plasmid, and then you coax them through tissue culture into a mature plant. And so every cell, the roots, the leaves, the branches, every cell that's living should have the gene of interest. In this case, they chose herbicide, but they've done this a lot with drought resistance too. They're looking at that pretty carefully. And it works. You put that plant out in a drought resistant or drought prone area and it survives. So these types of techniques have really been so powerful in the plant science realm of trying to help plants deal with different abiotic stresses as well as biotic stresses like certain pathogens. And they've had really good success stories. And so here's just another way to look at it. In this case, what they did was they're looking for disease resistance in this plant. So that plant is susceptible. They want to make it disease resistant. And so again, they took a disease resistant gene from a different plant, put it in to the agrobacterium. They took a marker for, in this case, they used antibiotics. So what they do is they put antibiotic resistance gene in there, as well as the disease resistant. And then they, again, they go to the tissue culture, mix in the plant cells, the bacteria, they all mix together. They start, agrobacterium affects the cells. And only those cells that have the plasmid that have the antibiotic resistance, you'll flush it with antibiotics, and only those cells that took up the plasmid will survive. And you'll be able to work with specifically the ones that captured, got transformed with that plasmid. So you just have to have some way to figure out which ones got the plasmid or not. So that's why you can do these, in this case, they added an antibiotic into the mix. And so it's pretty straightforward. They have a lot of protocols out there already, like troubleshooted and good to go for working with different, especially commercial crops that we use growing food. And so this is something that's really gaining in popularity because it's starting to get easier and easier, and the expense is coming down. Questions on this idea of using agrobacteria? Kind of surprised. Yeah, go ahead. No, yeah, it's part of the plasmid, and it'll stay part of the plasmid, but it doesn't interfere with what you're trying to accomplish. Mhm. Usually plasmids are big enough where you can fit like 9 to 12 genes on there and get it to work just fine. And you can order plasmids, right? Like companies, they have like these huge long databases of different plasmids you can order. Yes, that's a very good point. So when the plasmid is put into the plant chromosome, let's say, depending on where it gets put in, it could create a problem. Yeah, yeah. And so they have to, troubleshoot that. And so a lot of the protocols that they already have up and running, they work through these details, important details. Yeah, for sure. Yes. Well, yeah, it's still in the stage where it's usually done by companies that are doing the molecular biology aspect of it, and then they'll sell the plant seed from that, and they'll say, this seed's been, or this plant, these are seeds from a plant that's been, engineered to resist disease. Yeah. Like, like you put like a gene into one plant that like kills bugs, but then it gets into a farming plant. Couldn't that affect the ecosystem? Yeah, that's another thing that they have to look at with. So, can there be jumping, so to speak, into other species or impacting other species that weren't intended, And that's something, there's been cases in the past where trouble has come about because of they didn't think it through far enough. So the laws right now, and they're they've become more stringent, more tight on what they're allowing and the proof that you have to show of have you thought about what could happen? And so they're being more careful about that. A couple decades ago, it was more like just do it and throw it out there and it's good to go. And now we're realizing, yeah, well, we need to think about this and this and this. So there's a lot of usually greenhouse studies done in a controlled environment where they can play around with that and make sure that they minimize risk for sure. Yeah, that's a concern. Yes. Well, I know with herbicide resistance, there's been some issues where that trait has jumped into nearby plants in a nearby field, so to speak. And that I can't get into that now because it's a whole long story. It's pretty involved, but it was a pretty contentious, from what I remember, it's not a very good story. So yeah, and I think again that's like a case study going. We need to think about this more carefully. And then what came about during that whole drama? It was kind of there's a lot of politics involved in that particular case study that I'm thinking about. So, yes. Once you get used to out into a field process, you have to have like a certain critical of them with that you change to ensure that like the following generation from opportunity. Like, are they going to section with other things that are not in area? It's potentially, yeah. Right. Yeah. So then that would be the case of tracking it into the next generation and seeing what happens. And some of the research that they do with this, they do look at that, like, does the trait get transferred into the next generation? So that is something that you definitely have to think about. Yeah. Yep. But they've been looking at this for, like I said, drought resistance, disease resistance, flooding. They've got some good results with rice and flooding, weirdly enough, but you don't want to overflood your rice plants. And then, yeah, so there's a lot of different studies looking at it. And then going back to the endophyte idea, there's a lot of people that are saying, well, let's look at the endophytes to help the plants deal with this. And so there's more than just this avenue to help plants deal with their challenges in their environment. The endophyte research is really gaining in popularity because it's not genetic modification. It's just looking at mutualistic interaction, which is really cool. So lots of different ways to approach how do we help the plant. Okay. So besides using agrobacterium, even though agrobacterium has been very successful, we say it has a broad host range, which means it can infect a lot of different species of plants, which is good in the sense of if you want to use it as a workhorse to get your gene of interest into these different plants, that's good, but it doesn't infect every single plant. And so they still need to think about other avenues. And so pretty much in other avenues that you might use, one of the goals is that you have to get large copies of the gene of interest. You have to figure out a way to build up large quantities of that gene because the success rate to get it into the plant is fairly low. So you have to start with a lot and work with there. And so if you've taken BI-218, you might know about the idea of the PCR, polymerase chain reaction, making many, many, many copies of a section of DNA that you want copied. So that's certainly a very valid mechanism. And then the other thing to do is use E. coli as a workhorse and make a bunch of copies for you. And so it's just using E. coli as a tool. So E. coli, you have millions and millions of those in you right now. And E. coli is probably the most heavily studied bacteria out there. And it is used as a workhorse for a lot of applied reasons. And so basically what you do, because we know how to work with it so well, it's very easy to say, okay, E. coli, I'm going to put my gene of interest in you. I'm going to put a plasmid with my gene of interest into you. And then I'm going to culture you, grow you up in the lab, make millions of you, and by association, millions of the plasmid. And then you can, cultivate out the plasmid. You get the plasmid out and you'll have a lot of copies of it. So that's another way to do it. PCR has kind of gained an interest because it's quicker. And you don't have the E. coli to worry about if you don't know how to work with it. And so PCR is another really, really strong option that a lot of people work with. And so basically you need a lot of copies before you can try to think of these different ways to get those copies of the plasmids into the plant. And so basically you're looking at kind of the sky's the limit in the sense of what are the genes you want to move into the plant. It can be through these processes pretty rapid. You don't have to do selective breeding and wait for the next generation to grow up and have them form seeds and all of that. You can do it very quickly, and you can transfer genes from wildly different species. So how do you do it? If you don't have agrobacterium to do it for you, one method is called electroporation, where you have plant cells that you're working usually in a broth liquid culture. You have the plant cells in there, you're shaking that around gently, and then you can apply an electric field to those cells. And in that broth, you have your plasmid, and you're trying to essentially weaken the cell wall of the plants well enough for the plasmids to get in, but not destroying the cell wall, obviously. You don't really want to do that. And so that electroporation, we don't do that in our BI-218 class, but that is a valid strategy. You could treat the plant cells with this chemical polyethylene glycol, and that also seems to help kind of prepare the cells for uptake of the plasmid. You can use a gene gun, and it actually looks like a pistol. I worked with one when I was a grad student. I didn't do it directly. It was a fellow grad student who was trying to insert a gene of interest into a fungus. And fungi also have cell walls, so it can be really challenging to put genes into a fungus too. And so he rented a gene gun from another university. It was pretty cool. I'll show you a mock-up of it. And it worked. So another idea is to inject your gene of interest into protoplasts. So protoplast, I'd like you to know that word, please. That's a common term. So that is those are just plant cells that have had their cell wall removed. So no cell wall. You have to be very careful, but there are tried and true recipes out there where you can expose the plant cells to certain enzymes and chemicals to strip the cell wall, but leave the cell membrane intact. And then we say they're protoplasts. Now, in that manner, when they're in the protoplast stage like that, it's much easier to get DNA in because you don't have that cell wall to deal with. Okay. So microinjection, and you probably have seen video of that, where like with, when they inject sperm into an egg, it's kind of that idea, where you use a very fine needle and kind of do surgery. I'll show you a picture. And then you could have your gene of interest, and you essentially wrap that gene of interest with a liposome, which is just a fancy way to say it's a phospholipid bilayer that you use to kind of encapsulate your gene of interest. And then you mix that in with your plant cells in a liquid culture, and you let them mix and mix, and then the liposome will fuse. They usually use protoplasts for this. The liposome and the protoplast cell membrane will fuse, and then the gene kind of gets dumped into the plant cell with that fusion. And so there are these different strategies that have been employed. Some are easier than others, and then it depends on the plant species, which one you might opt for in terms of what's been done in the published arena to see, you try to not reinvent the wheel. If someone has figured out how to do one of these methods, you try to go with the tried and true methods. But here's the idea of a gene gun, and it actually looks like a pistol. You had it clamped up provided in a sterile environment, and you have a plate of, in this case, it was plant cells, but when I saw it, it was fungi growing across the plate. And then you literally shoot the DNA at the plant cells. And what they do is they have little gold, literally gold pellets that they coat with your DNA. And then they shoot it into the plant cell. And it works. Yeah. Yeah, I don't know. That's a really good question, because you'd think that'd be kind of an irritant, for the plant. So my thought is that somehow they extract those out somehow, but I don't remember. And I'm trying to remember when we did it for fungus. I can't remember what we ended up doing with the gold beads. But it's a very high propulsion. I mean, it does sound like a gun going off, when you do, and you literally have a trigger. And so there is kind of this brute force, get it into the plant cell. And if it's, fast enough, it can work. Here's the idea of microinjection. Oh, I'm sure it does. Some cells get damaged and die. Yeah, so it's not 100% by any stretch when they do the gene gun idea. I don't know. They might have changed it by now. They might have changed it by now, but that's what they started with. Because gold is not something that tissue usually reacts to. That's my understanding why they pick gold. Like it doesn't cause disease or irritation, I guess, to the tissue. So I'll try to see if I can find a protocol and see what they've done and if they still use the gold or not. Here you can see the needle coming from the right there, injecting into the protoplast. So that's what protoplasts look like under the microscope. They go spherical like that. I had a student do a directed study research project, and he was able to get protoplasts. We followed a couple different recipes. Yep, yep, yep. Everything's there. So it works. You can get them to form. The problem is I wanted to do it in lab for this class, but the problem is it takes like, I don't know, 6 to 8 hours. And so we only have a 3-hour lab. And so it doesn't really fit into how we run our labs right now. So I haven't been able to figure out a way to get it in. But anyway, but all of these methods, here they're showing protoplast again. All of these methods, you tend to work again with plants and tissue culture, so that once you transform your cells, however you do it, then you coax them in the tissue culture strategy, or coax them into a full plant. And so you have to do that tissue. And, depending on what plant you're working with, you can take different plant cells. I've seen papers written where they do even flower tissue. They work with flower tissue. So it just depends on the plant and what they're trying to accomplish. So like I said, they've been using these various techniques. And again, looking at how do we improve plant growth overall. And with crop production, a lot of it is we want them to withstand herbicide sprays so that when the weeds pop up, we can spray the fields and get rid of the weeds, but have our crop of interest still growing. So that has been probably one of the more famous examples of this kind of technology. But like I said, this isn't the only avenue or the only idea. There's lots of other ways in which they're using genetic modification. Here you're looking at papaya plantation, where the trees in the background are all susceptible to the virus. The ones in the foreground were treated through genetic modification. And so plant viruses can be devastating. We've already lost a couple rounds of our bananas, the type of bananas that we grow commercially. They've been wiped out twice just within the last several decades. So the bananas you eat are very different from the ones your grandfather ate or grandmother ate. So, and that's because of viruses sweeping through the plantations. Here's a nice visual showing the idea of putting in extra genes for a sodium ion pump, which basically lets the plant pump out more of the sodium that might come into their cells. They're more effective at pumping it out, so they can live in really salty environments, which tend to be also drought-prone environments, and do quite well. And so the plants on the left are a control, right? A is near zero in the pots labeled A, 50 millimolar in B, 100 millimolar of salt in C, 150 in D, and 200. So the top image are ones that have not been transformed. You can see when you get higher levels of salt, it's not doing well. But then you put in extra copies of the gene through genetic modification, and you can see they do quite well. So in really salty areas, I mean, we're running out of land to grow plants, like the good land, the good quality soil. We're running out of it. And so this is something they're looking at. Can we grow plants in more not so great soils, like really salty, drought prone, too sandy, and so they're looking at these ideas and they're they're making progress. It's encouraging. And also, if you think about it, stuff like this, like how do you ship tomatoes that are all ripe and not have damage by the time they get to the grocery store shelves? It's like, how do you do that? And so often they want to ship them green and then ripen them at the area of destination. And so, um, and then can we get them to resist spoilage? So spoilage is a big thing. That's something that doesn't really get talked about very much in like ordinary things when we think about how to feed all these people. Once you harvest the plant, that's half the battle. Getting it to grow to maturity, okay, great. Then you got to harvest that and store it before it can get shipped out. That's a whole nother avenue of things that can go wrong. And so it can be very, very challenging. Farming is one of the toughest professions out there because it's super challenging to get that product all the way to the grocery shelves successfully. All right, here's a wild idea. Plantibodies. So we actually use mice for this. So here's this weird thought. Okay, so plantibodies, what are those? They're antibodies. I abbreviate that AB. They're antibodies made by animals, usually mice, against a particular plant pathogen. So let's say you take that mouse, and basically what you're doing is you expose the mouse to the pathogen. The mouse starts making antibodies. You figure out what gene got turned on to make that antibody, and then you grab that, put it into the plant, and it apparently works. And so you can actually get, what they do is they get the plant transformed and then they expose the plant to the same pathogen, and they actually see that the plant has the antibodies necessary to defend itself. Pretty weird. I don't know how common this method is. I found it in a publication. I haven't really seen it much out there in the published literature. I don't know much about it. We're like stuff that affect us. Mm-hmm. Yeah, they don't really put the animal cells in there, but it's more the gene or yeah. Mhm. Yeah. But yeah, I mean, it's a good thought. It's like, do we need to worry about that kind of stuff? And yeah, absolutely. That all has to be thought out really carefully, and do your due diligence on everything. So, um, and maybe that's why this really hasn't, I don't see it out in the literature that much. I can do another search over the weekend and see if anything more has popped up on this. But I really don't see this as a method when I look at how they modified the plant. But like I said, I found one publication on it where it was a thing. And so I was wondering if this was going to take off or not. Okay, so, um, thinking about commercial crop production, all the different plants that we grow, and how that might, we use plants, of course, to eat and for health reasons. But basically, when we look at how to grow these plants successfully so that we end up benefiting from that, they really are looking at helping the plant defend itself better against pathogens. And of course, that's going to get you a better crop yield. Here's a really important idea that's becoming more critical: increasing the nutrition per gram of plant tissue. And so, like I said, we're running out of land to grow crops. So if we want to feed more people, another way to look at it would be increase the nutrition per gram, so that per gram of tissue, they're getting more good quality nutrients, and then they don't have to eat as much. And that's something that people are really starting to look at more carefully as part of the way to solve this. How do we feed 10 billion people by the year 2050? And so basically trying to find ways to increase crop yields. They're making good progress with rice. In fact, they're trying to do genetic modification to get rice to do a certain type of photosynthesis called the C4 method, which you'll learn about shortly. And so the C4 version of photosynthesis is really efficient. And so they're trying to ramp up rices, like get more efficient so that they can get better crop yields. Rice is a very, very important crop. They're looking at drug production in plants as well. Edible vaccines, that's something, if you think about third world countries where you have to deliver vaccines to people that don't have refrigerators. How do you keep the vaccine stable? And so one idea is, well, don't deliver it that way, get it into the food they eat. And they're actually doing that with potato tubers. They're trying to modify potato plants so that the tuber that gets produced has the necessary edible vaccine in it. So that's something that they're looking at. Vitamins and minerals, of course, that's kind of like increasing the nutrition per gram of tissue. They're really focused in on, a lot of people are iron deficient in third world countries, and so they're looking at that idea, beefing up the protein per gram of rice tissue. Vitamin A, you might have heard of golden rice. That's been a genetic modification to ramp up vitamin A, which there's a lot of severe vitamin A deficiencies in third world countries. Salt tolerance we mentioned, and then looking at phytoremediation. So that's the idea of using plants to clean up toxins in the environment, primarily in the soil. And so through all the human-made activities, we've managed to pollute a lot of different soils. And so phytoremediation is the idea of using plants. They call it the green clean method, because it looks nice as they're growing on that contaminated area, but as they're growing, they're busy detoxifying that region. So not really just the plants doing that, by the way, it's the plants and their associated microbes that are doing it. You can't separate them. And so it's actually bacteria, often fungi, hanging out with the plant are actually the ones doing the lion's share of the remediation, but it helps to have that partnership. If you get more toxin breakdown. Yes. Yes. Some of them are endophytes, you bet. And then some of them are also just microbes that hang out like right near the roots. So they're not in the plant, but they're like hanging out with the plant. And so, but endophytes too. They're finding endophytes definitely. In fact, yeah, there's some really cool studies, in fact, where they did work with poplar saplings, and they had endophytes that they took from a different plant species, and they put I think it was toluene degradation or benzene degradation gene into that, and then they mixed it in with the poplar endophytes, and they could show that bacteria put the plasmid into the poplar endophytes. And so it's like they were able to get that trait into the endophytes of the poplar, which was great because, they had found this endophyte. They're like, oh, this endophyte can really break down benzene, but it hangs out with this species, not poplar, and so they're like, could we put it into poplar? And what they found out was kind of like it went into poplar, but it didn't persist, but it went in and persisted long enough to share its genes with the endophytes. So that's, to me, super exciting. That means you don't have to have the perfect endophyte that can hang out with the perfect plant. You can have this plasmid transfer going on. So really cool stuff with looking at using endophytes for remediation. We get a lot of toxins to clean up in our environment. When I teach advanced botany, we look at the map of the United States of where all the toxic sites are. Oh, it's scary. And I'm sorry if you live in California. There was so many hot spots in Central California that we were looking at in that particular study. But yeah, it's across the nation. It's scary. Okay, so if you are thinking about putting a gene that does not, come from the same plant or the same organism, and you want to get that gene into the plant through genetic modification, like we've been talking, it's possible. Often, what they've done is they can take also protoplasts from different plant species, and so this is very artificial. What they do is, so they say, well, these two plant species can't produce viable seed because they're different species, so they can't go through, they can't have sex with each other, right? So, well, we like the traits in this one and we like the traits in this one. It'd be great to create a plant that had both those traits in a single plant. So how do we do that? They tried this and it actually worked, at least with some of the case studies. They take the protoplast, so they strip off the cell wall of those two plants, and then in the lab, in usually a liquid culture, they mix them together. And what happens is they will mix together, and then you usually apply an electrical current or polyethylene glycol or something like that. And the two cell membranes of the protoplast will fuse. So you get a cell now, and with the mixing of the DNA in that cell. And so protoplast fusion actually has worked. And then they start referring to them as hybrid fusions at that point if it's successful. And you can take those and then coax them to grow into a full plant through tissue culture technique. And once that occurs, then they are called somatic hybrids. So these are the plants that result from this are called somatic hybrids because you didn't use sperm and egg, because you can't, because they're two different species. So we can't get this sperm and this egg to work. It won't work. But so somatic means body. It's not non-sperm cells, non-egg cells. If you say it's a somatic cell, it just means it's normal body cells. So pretty wild. But again, plants are pretty amazing. And so they did that, and this is the result that they got. Okay, keep thinking. So on the left is Indian mustard, and on the right, I have it in the notes here, stonecrest, and then the hybrid in the middle. And so has to do with their tolerance to heavy metals. So it says that the one in the middle has high levels of lead, zinc, and nickel. So this represents a wide cross between plants. This crossing was done by protoplast fusion. Indian mustard tolerates lead, but not zinc or nickel, whereas stonecrest tolerates zinc and nickel, but not lead. The hybrid plants do all three. So basically, you took traits from each of those and created a super plant. You mean like if this plant produces seeds? Is that what you mean? Or like if one plant has colors, like yeah. Yeah. Mm-hmm. Yeah, it's pretty fine too. So the middle plant even though it is technically not the same function of those of its previous. Right. Yeah, I mean it's like a artificial. Yeah, really, right? Because you took body cells from the left plant, body cells from the right plant, and created it. So I mean, you call it a hybrid. How they're referring to those, I don't know. Like, how do they? What jargon are they using other than hybrid? I don't. What are you doing? Yeah, exactly. Very good question. I don't know the answer to that one. All right. So remember to do your lab homework. Please do that by Wednesday at 11. Okay. We'll stop there for today. Have a good weekend, everybody.